Second white coat. Subscribe and like. Let's learn medicine together.
All right, let's dive into a medical mystery that plays out in the crucial hours and days right after a stroke. It's a story all about brain swelling, but as we're about to find out, not all swelling is the same. Not even close. So this is the clinical note at the very heart of our puzzle. Imagine this. A patient has a stroke, and they seem to be getting stable. But then, a few days later, they suddenly take a nosedive. The reason? Worsening mass effect. That's just a way of saying the swelling is getting so bad, it's literally crushing the brain from the inside. But the real question, the one we're here to answer, is why the delay? Just look at this timeline. Day 0, the stroke hits. For the next 48 hours, things are looking okay. The patient is stable. You'd think they're on the road to recovery. But then, bam, day 3 hits and they suddenly get confused, drowsy. Their condition just plummets. What on earth is happening inside that brain on day 3 that wasn't happening on day 1?
To solve this mystery, we've got to rewind the clock. We have to go back to the very beginning. The first crisis kicks off within just minutes to hours of the stroke, and it's a story of pure cellular chaos.
This first type of swelling has a name, cytotoxic edema. Now, the key word here is cyto, which means cell. The fluid isn't just leaking into the brain tissue. No, it's being pulled directly inside the individual brain cells, causing them to swell up like tiny overfilled water balloons. So how does this even happen? Well, it's a disastrous chain reaction. See, the stroke cuts off blood and oxygen, so the cells can't make energy, ATP. And without that energy, these tiny little pumps that keep the cell's salt balance in check just fail. Sodium floods into the cell, and you remember this from high school biology, right? Water always follows salt. The result is a swollen, dying cell. And we're not just talking about a few isolated cells here. The brain's entire support system is under siege. Your neurons, the cells that do the thinking, they're swelling. So are the astrocytes that support them and the oligodendrocytes that insulate them. It's a total local system failure. So if you want to boil it down, cytotoxic edema is simple. The pumps die, the cells swell up, and you get an early spike in the pressure inside the skull. This explains what's going on right after the stroke, for sure, but it still doesn't solve our mystery of the delayed decline. For that, we need to look at the second wave. Okay, let's move on to Act 2 of this story. This part doesn't even begin for another 24 to 72 hours. This is the delayed threat, the second wave of swelling that finally explains what went wrong with our patient on day 3. The second type is called vasogenic edema. Vaso means blood vessel. So now the problem isn't fluid getting pulled into the cells. The problem is that the blood vessels themselves have sprung a leak, and fluid is seeping out of them into the space around the brain cells. To really get this, we have to talk about something called the blood-brain barrier. I mean, think of it as the brain's ultimate security fence, right? It's incredibly strict about what it lets in and out. Well, in the aftermath of a stroke, that fence gets breached. The gates are just busted wide open. So what breaks down this fortress? Well, that initial stroke triggers a massive inflammatory response. The body just unleashes a flood of chemicals, including this one thing called VEGF, which actually makes blood vessels more leaky. So now you've got this protein-rich plasma oozing out into the brain tissue, and you guessed it, water follows right behind. So what we have here are two completely different enemies, attacking the brain on two totally different schedules. Let's put them side by side. It'll make the difference crystal clear. Okay, check out this breakdown. Timing: cytotoxic is lightning fast, minutes to hours. The blood-brain barrier is totally fine, and all the fluid is stuck inside the cells. Now look at vasogenic. It's the slow burn, peaking days later. Here, the barrier is broken, and the fluid is outside the cells. This also explains why steroids, which help patch up that leaky barrier, only work for vasogenic edema. Look, if you remember only one thing from this whole explainer, please let it be this. Cytotoxic fluid is inside the cells because the pumps are broken. Vasogenic fluid is outside the cells because the barrier is leaky. That simple difference in location, that's the key to this entire mystery. So it's time. Let's put all the pieces together and finally solve our puzzle. We can go back to that patient, stable on day 1, crashing on day 3, and now we actually have the answer. After seeing these two completely different timelines and processes, what's the one crucial piece of information that just cracks this whole case wide open? And there it is.
The patient's terrifying decline on day 3, it wasn't a brand new problem. It was the peak of the second wave. It was that vasogenic edema, driven by inflammation and a leaky blood-brain barrier that caused that incredibly dangerous delayed spike in brain pressure. The mystery is solved. And that just leaves us with a really fascinating final thought. We've just seen that a huge, life-threatening part of the brain's response to injury can be delayed by days. It really makes you wonder, doesn't it? In medicine, in recovery, and life, what other critical processes are we only ever seeing the very beginning of?